Welcome to the Advanced FSL course lecture on writing data processing pipelines. In this lecture and the associated practical, we will look at how we can combine all the tools that we have learned about in the main FSL course and in tools from any other neuroimaging library and see how we can use them to write pipelines to process our own data. But first, let's define what we mean by a data processing pipelines. Well, during the practicals, we saw that there are a lot of individual steps to processing our data. For example, if we have structural MRI data, we might want to do brain extraction, bias field correction, segmentation, registration, and only then can we move on to VBM or vertex-based analysis. Functional data has its own set of steps, diffusion data, and in these advanced course practicals, we can also see the individual processing steps for ASL and MR spectroscopy. So we have to run all of these processing steps, not just for one subject, but your typical study might have tens of subjects or even hundreds of subjects, and rerunning all of these steps by hand from the command line might be very tiring. There's just a lot of individual commands to run, and there's a lot of room for mistakes if you start to try to rerun these commands for each individual subject individually. So ideally, we would tell the computer which commands you want to run to process the data of each subject and in which order you want those commands to be run. And that is basically a data processing pipeline. It's telling the computer how to process the data for each subject so that you don't have to. However, a very important part of running any data processing pipeline is that you do still want to visually check all of the outputs to see that all of these individual checks actually worked for each individual subject. And we saw during the course some great fossilized commands which would allow us to interactively view how, for example, in this case, the brain extraction step has worked for our field map image. However, if we start rerunning the same fossilized command across a lot of different subjects, and for each step in our processing pipeline, again, it becomes very cumbersome. So, ideally, we would also like to efficiently visually check the outputs across multiple subjects without having to rerun a lot of individual fossilized command. So, how do we do that? How do we rerun our processing steps across all these subjects? Well, one way to do that is actually running a pre-existing pipeline, and you've actually done this before already within the FSL course, such as, for example, FEAT is a pre-existing pipeline which was written by us to basically process your functional MRI data and to run the statistics. It does pre-stats, it does registration, statistics, and some post-statistics analysis. And in all of these things, you have a bit of control about how all of these individual steps are run. And then we can visually check the output using the resulting HTML files. Even if they're not interactive, they do give us an idea of how well, for example, the brain extraction and registration worked. However, if the registration didn't work, for example, because you've got the subjects with large lesions, and you would like to mask out these lesions when running the registration, then that is possible within the individual tools. For example, if you would run FNIRD, you could mask out individual lesions and get a better registration, but that actually isn't available within the broader FEAT pipeline. So we can't actually run FNIRD in a way within the FEAT pipeline that does this. So you would still have to write your own registration, do your own registration step, and then feed that within FEAT, which quickly becomes more complicated again. So ideally, we would like to easily write our own pipeline, which actually processes the data in the way that we want to process it, that is ideal for our data set, where we correctly deal with the peculiarities of our data. And we don't necessarily want to be limited to FSL. We want to use multiple neuroimaging tools. And of course, we want to structure our data in the way that we want to structure our data, which might be the BITS format that you might have heard of, which is a standard way of structuring your data, which makes it easy to share across projects, but it might also be that within your lab you use a different way of structuring your data, and ideally the pipeline would run irrespective of how you structure your data across multiple subjects and be flexible across different data sets. And finally, we want to have visualization of the pipeline results efficiently across multiple subjects. So within FSL, we do include two tools which help to create such pipelines. So we've got FileTree, which helps you to describe the directory structure both of your input data and the resulting output data from, from your pipeline. So in this case, so here on the top, we've defined what our input data looks like, which is apparently supplied to us within an input directory, in which case we have within which we have subject-specific directories, some of which at least have multiple sessions, and then we have this total final file, which actually somewhat closely follows the bits formatting. And then we've got the output for, in this case, an FSL VBM-like pipeline. So we run brain extraction, we run segmentation, we run some segmentation, and then we create a template as part of our FSL VBM analysis, which we will look at more closely during the practical when we will actually create this pipeline. So the next step from defining where all of these files will be placed is to actually define how these files will be produced. So this is where the second tool, FSL Pipe, comes in. And so FSL Pipe, you will just tell how to produce a T1W brain file from a T1W, which is by running FSL Bet. We will tell it how to produce the gray matter segmentation by running FSL Fast, how to run registration by running FSL Nerd, and how to create a template from the individual files by running FSL Add. So we basically just define all of these individual steps, and then FSL Pipe will put that together into a pipeline which will run across the multiple subjects, in this case across the multiple sessions, and produce the appropriate template, as we will see in the practical and in the remaining two talks, where I will discuss in more detail first FileTree and how we can use FileTree not just as an input to our pipeline, but also to visualize our pipeline results within Fossilize. And then in the third talk, we will look at actual how we can use FSL Pipe and minimal programming within Python to actually define all of these individual jobs and get a fully functional data processing pipelines. See you in the